In this video, we are going to see about the instrumentation of centrifugation. Already we see the centrifugation process principles. Here we are going to see about the centrifugation instrumentation. A centrifugation instrumentation is going to depend on the centrifuge tubes, rotor and motor. This centrifuge tube is going to depend on the rotor types. So at first we should know what are all these means. So the centrifuge tubes is going to, all the centrifuge tubes are going to depend on the rotors, but in common all the centrifuge tubes has a conical bottom. These conical bottoms helps for the sedimentation of the biomolecules and decreases the collapsing of biomolecule sedimentation. It is very important for the centrifugation process. Then rotors. The rotors or the rotating unit. These rotors are going to have a central axis which is going to arrange the tubes to this central axis. So the centrifuge rotors or the rotating unit which has a central axis and have a fixed holes with an angle to hold the centrifuge tubes, which helpful for sedimentation. Another important thing is motors. So the motors are going to cause the running of rotors. The rotors are going to involve in circular motion which is driven by the motor. So the centrifugation process is going to be started only by the motors. So the sedimentation is going to be taken place only by the motors, which causes a centrifugal field. So the motor is important for the production of centrifugal field, where the rotors are involved in arrangement of test tubes which involved in sedimentation. So this is going to be a centrifuge which has a rotor. This is going to be a rotor which has arrangement holes for the test tubes. These are test tubes. And motor, here is a motor which is going to drive this central axis rotor. So these are the instrumentation of the rotors, centrifuges. So the three important things to be involved are rotor, motor and the centrifuge tube. So these rotors are going to depend on the arrangement. Next, we have to see individually different rotors. So first is going to be a fixed angle rotors. From this name itself we can tell that fixed angle rotors are going to have a fixed angle. This fixed angle is going to be 14 to 40 degree. 14 to 40 degree is the angle that is fixed for the fixed angle rotors, which is going to be slightly outwards from the axis. So these fixed angle rotors are arranged at the certain degree of 14 to 40 degree. It has a rotor speed of highest amount but decreased in the zonal rotors, but among the other three it is going to be increased in RPM, that is revolution per minute. That is, a revolution of this rotor is going to be 1 lakh per minute. It is going to be revolve about 1 lakh per minute. So the capacity of this centrifuge, which is going to be inclined at the 40 to 40 degree of test tube, is going to have the 0.2 to 1 liter of sample can be accommodated in this centrifuge tube. So when the rotors are going to spin, that is revolve, it causes the biological particles to move radially. That means this is the central axis. So this is going to be a centrifuge tube. This centrifuge tube is going to be at the fixed angle of 14 to 40 degree. So when it is going to be revolved, during the rotation, the biological materials are going to move radially, that is in the circular motion. This circular motion causes, when it is decelerates, there is no cause of the change in the angle, that is fixed at 14 to 40 degree. This radial movement of biological material going to hit on the walls of the test tube. In the test tube walls, these materials are going to hit due to the radial movement. These hitting of these molecules into the walls of the test tube, which causes the material to slide down and sediment at the bottom. This is known to be a wall effect. So, in the fixed angle rotors, there is an important principle or action to be occur is known as wall effect, where the particle is going to be moved in the radially outward direction for the short distance and strike and slide down on the wall and pellet out. This is going to be an effect to be occurs. After the sliding down, it is going to be a sedimentation quicker due to wall effect. When the revolution is going to be stopped, there is going to be a pelleting out or sedimentation occurs. The advantage for this sedimentation or fixed angle rotors sedimentations are found to be involved in isopycnic centrifugation and density gradient centrifugation, where there is a different components are going to be separated off, because each components are going to have a different viscosity and thickness. Based on this, a density gradient centrifugation are going to be occurs with these fixed angle rotors. A major disadvantage to be occurs in this, these rotors are going to have a lack of organelle separation. So, it is involved in the density gradient centrifugation, which is going to be involved in the sedimentation of the compounds based on density, but a differential sedimentation or not occurs. In differential sedimentation also going to be takes place based on the density, but it is going to be depends on the density of the biomolecules. That means a different biomolecules can be sedimented off by differential centrifugation, but in the case of density gradient centrifugation, a different components are going to be separated based on the density of the medium. A different medium is going to be used, which causes the sedimentation to occur. So, a different mediums can help with the fixed angle rotors sedimentation, but a different organelles cannot be separated by the fixed angle rotors. So, this is going to be an image where how does this centrifugation occurs. This is a biomolecules which is going to move radially and which causes a biomolecules to slide down and pellet out when there is a centrifugation stops. Next, we are going to see about the vertical type rotors. In the name itself, we can found that a vertical angle rotors are going to have arrangement or holes for the centrifuge tube in the axis, along the axis of the central axis. So, these rotors are about 90 degree, that is going to be a angle of central axis. This is equivalent to the central axis angle, but the fixed angle rotors are going to have 14 to 40 degree. Here it is going to be 90 degree. So, it is going to have a rotor speed which is decreased level than the fixed angle rotors, which is about 50 to 65,000 rpm. That means a revolution of these rotors are going to be 50,000 to 65,000 per minute. The capacity of the centrifuge tube is going to be 0.2 to 1 liter, which is equivalent to the fixed angle rotors. So, here a small amount of wall effect to be occurs, but in general the wall effect is not prominent. Here the centrifugation is going to be occurs based on the biological particles based on the biological particles test tube. So the compound is going to be moved radially along the walls of the test tube, which is going to be on the surface of the test tube during the acceleration. When there is a deceleration to be occurs, that is when the centrifugation is going to be stopped, the compounds or biological materials to be involved in sedimentation, which moves on to the test tube surface radius along the wall of small amount, are going to orient within a shorter time to the pelleting out. So the small amount of wall effect to be occurs, but it was not prominent, because here the sedimentation is occurs because the compounds are going to move along the surface of the particle, but it was small amount of wall effect which causes the sliding down during deceleration. So the main advantage of this pelleting out is for DNA, RNA and lipoproteins, where it is going to have very fragile. So this centrifugation causes the sedimentation, which is not harsh wall effect to be occurs. The major disadvantage in this pelleting out is fallback of solution, where here the compound is going to be sedimented here. In certain times before the deceleration itself, these compounds get falling off and get mixed to the medium, and again we have to undergo the centrifugation. But on the fixed angle rotors, the sedimentation is going to occur along the wall and slide down, so there is no this types of falling back to be occurs. So this is a sample. The sample get pellet in the radius of the test tube as a small amount. After the deceleration, a small amount get fall back as very smoothly, and it causes pelleting out. Instead of pelleting out, it causes a different gradient also. Next, a swinging bucket rotors. In the swinging bucket rotors, there is no certain angle for the placement of test tubes. It is going to have a involved in large amount of samples to be involved in swinging bucket rotors. So there is no angle. It is generally a swinging. It is going to be swinging out in the horizontal position, which is perpendicular to the central axis. So it is perpendicular to the axis of the rotation, that is away from the central axis. The capacity is about 12 liters. The RPM is 4000 to 5200 RPM, which is very lesser than the fixed angle rotors and another rotor. And the vertical tube rotors. So this 4000 to 5200 RPM is very important, which means there is revolution to be occurs is 4000 to 5000 times per minute, which is very lesser than the both, which means it's going to have a decreased time. Within a few seconds it's going to undergoes the sedimentation. So when the acceleration is going to be occurs, the test tubes are going to orient as perpendicular to the axis of rotation, but it was going to be parallel to the centrifugal field. When the centrifugation stops, the rotor decelerates. The tube is going to fall at the original position along the axis. So during this period, that is going to be away from the central axis along the centrifugal field. During this period itself, there is a different types of compounds is going to be sedimented off based on its density. So it is in general, it is going to be involved in density gradient centrifugation, which means different compounds, that is different components or cell components can be separated off in this swinging bucket swinging bucket rotors. It causes a cushion effect, so it lacks the wall effect, which causes a harsh to the components, which causes the mixing of components. A wall effect causes this, but it lacks the wall effect and causes smoothened effect, that is cushion effect. Then a major disadvantage in it is lacking of pelleting. So when the different cell components are going to be recovered, the recovered components can mix up, because it is going to form the components in different region based on this density. So when we going to recover these individual components, it can undergoes mixing. So it is a major disadvantage. It also causes the swirling effect, which causes the mixing of separated components. Then zonal rotors. Zonal rotors are going to be used in industrial purposes, where there is large amount of components can be separated off. So this is a 4 sectors which is going to handle different samples, where there is samples can be loaded with these zones. So during centrifugation, the components can be separated in different zones based on their density, where we here can see these are all one cell components, these are all one cell components, these are all one cell components. So based on their density, they form the different zones. Here we can see a vertical zones of its separation after the loading. So these zonal rotors itself taken place in the two methods. In general, the zonal rotors are going to have the sectors which is 4 equal quadrants. The rotor speed is going to be 5000 to 60,000 rpm, which is higher than the fixed angle rotors and vertical fixed angle rotors and swinging bucket rotors and other. So the maximum capacity is going to be 50 to 100 times more than the swinging bucket rotors. So it is going to handle more amount of samples, which is used in industrial purposes. It is of two types, batch type and continuous flow type. In batch type, there is going to be loading of samples batch wise, but in continuous flow type rotors, there is continuous sedimentation to be occurs by the sample. After the sedimentation of the sample, there is continuous injection of another sample. There is no amount of batch type rotors like to be happens. It is going to occurs continuously. There is continuous flow of samples to be occurs. It is in batch type rotors itself, it can be a low speed or high speed rotors. There is a loading of gradients followed by running of rotors. But in continuous type rotors, the gradients are load without any rest. Without any rest, it is going to be flowed permanently and continuously. In general, it flows about 2000 rpm, which is decreased level, because it is going to be occurs or running out continuously. The major advantage is going to be it involved in minimizing the wall effect, but it is going to decrease the wall effect, and sample size is going to be increased, which is very important during industrial purposes. It is also involved in cells and viruses sedimentation, because it decreases the wall effect. It removes the contaminated proteins, hormones and enzymes like macromolecules too, because it has increased the sample size. So this is going to be a process how it occurs. Then the sample is going to be entered, it is going to be out, which means the samples are harvested off. In continuous type, the sample is generally after sedimentation harvested out continuously, and samples are injected continuously. But in batch type, it occurs in batch wise. So when it is centrifugation occurs, it gets harvested off like this. So next we are going to see about the types of centrifugation. There are about 4 types of centrifugation. So the types of centrifugation we can see in next video. Thank you.